So students, today we are discussing about the structure of a human testis. This is quite a simple structure. You just have to remember two or three things. Thus it. This is the basic structure of a human testis. So first of all, starting with this one, this tube-like coiled and subcoiled structure is called vas deferens. Okay, so after that, it connects with this long tube-like thicker structure. Okay, so this is called epididymis. 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 So you can just divide into three parts. This is the head region, this is body, and this will be the tail. So you can just write tail of epididymis. This is body of epididymis, and this is head of epididymis. Okay, so that's it. This is epididymis, and composed of, you can just basically divide it into three parts: the head region, the body, and the tail. Second, the ovoid structure, oval like structure, the main reproductive part of testis. This is it. So you can see two structure. It's just outer covering, this one, this whole thing, this one, and all over that whole thing. This is called the tunica albuginea. Okay, this part, and inside of it, this you can say globular structure, coiled structure, this is called seminiferous tubule. That's it. From normal section, from vertically, you can just have to mind four structures: seminiferous tubule, tunica albuginea, the epididymis part, and the vas deferens. And you can just divide the epididymis into three parts: head region, body, and tail. That's it. It's quite a simple structure. Okay, and after that we are going to the cross section. We if we just histologically cut it to a 6 to 7 micrometer diameter of it, then what could you see basically from this part, not the epidermis or the vas deferens. We are discussing about the main reproductive part of this. Okay, so students, today we are discussing about the structure of human testis. This is a histological section of 6 to 7 micrometers, and if you just stained it with hematoxylin and using stains, so you can see a colorful pictures of like violet and pinkish color of these structures. The nucleus part will be of violet, and the cellular parts here is all about like pinkish part. So what we can see after staining the cross section of the testis, we just see the testes are complex tubular glands entirely covered by a capsule. This is the capsule region. Okay, when a testis is cut, you can see this type of structures. This type of structure. There are so many of them. So this outer covering is called tunica albuginea. Albuginea. Tunica albuginea. Okay. So inside of it, we are just magnify this one to this whole thing. Okay. So we are getting it one by one. First of all, the outer layer, what you can see here, the outer layer is called the basement membrane. Okay, so this is the basement membrane, and inside of it there is mainly two types of cells you can see. First of all, these kinds of oval shaped structures. Okay, and second, this is like pyramid triangular like structure attached to the basement membrane, or you can say basement lamina. Okay, so these ovoid structures or oval shaped structures is called germinal cells. Germinal cells means from these cells the original spermatids or you can say sperms develop, and this is called the Sertoli cells. This bigger one, triangular one. So two types you can say. Number one is uh, germinal cells, and number two is Sertoli cells. The main reproductive part is this germinal cells. This is just a supportive accessory parts. So we are getting the shorter one first, the Sertoli cells. So from the basal lamina, we can see from the basal lamina or basal membrane to the inside of this whole thing, to the inside of this seminiferous tubular lumen. You can say this whole empty space here. This is called seminiferous lumen. Okay. So from the lamina to the lumen, you can say a triangular pyramidal structure just between the germinal cells. This is called the Sertoli cells. This also have nucleus as well, but the nucleus is not very distinct. It is kind of not circular, what you can see in normal cells. All right. What is the function of it? So this is the cell which provide food, nourishment and protection to the developing spermatozoa and other germinal cells. Spermatozoa means sperms, and other germinal cells means this kind of cells. We are getting it after that when we discuss it. So that's the Sertoli cells. Triangular shaped structure had a not distinct nucleus, you can say, and this provides food, nourishment and protection to the developing spermatozoa. Next, we are coming to the germ cells part. Okay, there can be 3 to 4 layers of these germ cells one by one. These are the laminar region has the biggest one, and gradually when you coming to the lumen part, it will just diminish its size and developing as a form of spermatozoa. You can see this one, then this one, then this one, and then the normal sperm or spermatozoa, you can see. Coming to the germ cells part, the first one is this part. You can say from the other big oval like structure, this is a quite different. In this case, the nucleus is not prominently developed. Okay, the the chromosomes inside of it just spread all above the all around the cells. So this one is called the spermatogonia. Okay, and after the whole nourishment part, when it got into the developing stage, it just compresses into a single composed nucleus, and that whole form is called the primary spermatocyte. Okay, next, it compresses in size. The nucleus is more distinct region, you can say. Through meiosis, the primary spermatocyte is dividing into secondary spermatocyte. Normally, what happens? The primary spermatocyte and just might meiotically divides and forms secondary spermatocytes. So these are secondary spermatocytes. So these secondary spermatocytes is divided mitotically, and the number of secondary spermatocyte sites is greater than the primary spermatocyte, uh, because obviously it is coming from the primary spermatocytes by mitotically dividing into secondary spermatocyte. So number of the secondary spermatocyte obviously increased. Next, the spermatids part, the primary spermatids. The other, when you come to the spermatids part, you can see two types of structure. Other one is here, here, and here, here. You can see a structure of this kind of structure, say oval shaped body with a tail forming of it. Okay, so this one is called primary spermatids, and next one is where you can see all above that region, this type of structure. This is mature spermatids. All right, so this is the whole structure. This is quite a simple structure to remember. Oh yes, a very important part, these are called Leydig cells. Okay, these are called Leydig cells, which you can say this is the endocrine part of the testis. Endocrine part means which secretes some kind of chemical transmitters, or you can say hormones. So they secrete male sex hormones called androgen. Androgen it's secreted due to influence of luteinizing hormone to the pituitary gland, right? So this is the whole process, this is the whole thing we are expecting with this structure. The germ cells, the Sertoli cells. Sertoli cells are for food production, for nourishment, and kind of protection from above. Next, coming to the germ cells. First of all, spermatogonia portion, nucleus is not composed quietly. Second, in the developing stage, it forms primary spermatocyte. It just compresses itself and form primary spermatocyte, not just like from the primary spermatocyte to a secondary spermatocyte. It just divides mitotically. But in case of primary spermatogonia to the primary spermatocytes, it just converts into it, right? There is no division between spermatogonia to primary spermatocyte. That's the basic. So after that, spermatocyte mitotically divide and form secondary spermatocyte. Then again, secondary spermatocytes develop a step-by-step process, uh, gradually by forming primary spermatids. Then the primary spermatids again develop to mature spermatids, and in between, you can say in between each seminiferous tubule. This whole thing is called a seminiferous tubule. In between two or three seminiferous tubule, there is always present a Leydig cell, which is the endocrine part of the testis. More of you think that the whole testis is secreting some this androgens. You can say testosterone or this kind of thing, but that is not correct. The correct thing is only the Leydig cell part can secrete hormone. All right. So that's the structure of a human testis. Quite a easy structure to remember. Just two parts to remember, and the developmental stages it all composed of. Okay.